sticking around for the final session of the day. So today I'm here to argue that given the dynamic nature of our planet, it's safe to assume that large-scale geological and climatic processes are important for diversification and community assembly. And as a result, to better understand regional and global biodiversity, we need to account for such processes. And for that reason, we stand to benefit from methods that allow us to look at contemporary genetic data from contemporary populations to infer patterns predicted by such past events. So for example, let's say we have a community of co-distributed species here that at some point in the past is split by some event. Black rectangle here, is as good as anything else. And then we go out today and we look at 3 co-distributed species of lizards that are distributed across this area. We might expect to find that the divergences of those species will be temporally clustered and coincide with when this event occurred. And we can think of this as a particular model of divergence, one that has a single divergence time parameter shared by all 3 of our population pairs, and so they all share the same divergence time. However, if we want to take a model choice approach to this problem, there's other divergence models that we have to account for. We can add a second divergence time parameter, and we can assign our first, second, or third population pair to that divergence time parameter. And so that's 3 more models. And then finally, there's a 5th model, which is the most general model that allows all 3 of our population pairs to diverge at unique times. So if we want to learn about the effect of this past event, it would be ideal if we could infer this divergence model and the divergence times from sequence data from these populations. However, in order to do that, there's other things we need to account for. So we're going to be collecting sequences from populations of organisms. So we have to account for the genealogical relationships of those sequences, as well as the mutational processes by which the alignments evolve along those gene trees, as well as the demographic processes that control how these gene trees evolve in those populations. So if we do account for all of that, we can take a Bayesian approach to this problem and try to estimate the joint posterior probability of those divergence times and other nuisance parameters, given our sequence alignments and a divergence model. And we can use Bayes' rule for this. The denominator of Bayes' rule is the marginal likelihood of that divergence model, which we'll get back to later. And if we sample over all possible divergence models, we can again leverage Bayes' rule to estimate the posterior probability of that divergence model, given the sequence data, which is essentially just this marginal likelihood of that divergence model weighted by the prior. So it's a lot of equations up there, I know, but all I want you to take home from this is this marginal likelihood is extremely important in estimating the posterior probabilities of the divergence model. Okay, so there's an existing method that does this, it's called MS-Bayes, and does this in an approximate-Bayesian framework. However, if we simulate data sets in which population pairs randomly diverge over millions of generations, and then we use this method to estimate the number of divergences, it will often tell us with strong support that there's been relatively few divergence events that explains the divergences among all these population pairs, even though they've been random. So that's not ideal because we're often interested in these sorts of models of co-divergence, and if the method is spuriously inferring them, that's not very good. However, I think the MS-Bayes approach is an elegant one, and so instead of abandoning it, my goal was to try to look for theoretical reasons why we might expect the method to be behaving this way, develop a new method accordingly that extends the MS-Bayes model, and hopefully allow us to improve our ability to estimate shared evolutionary models of shared evolutionary history. And so I thought of 2 possible ways of doing this, one being to use alternative priors on the parameters of the model, and the second being just taking a different approach to how the temporal distribution of divergences is distributed across our population pairs. So first, why alternative priors and why might they help this method? So here's our good old friend, the marginal likelihood again, which looks a little bit scary, but it's essentially just the likelihood averaged over the entire parameter space of the model. And it's a weighted average, it's weighted by our prior. Okay, so why is this important? Well, if we look at a contrived and simple example, where we're comparing the divergence times of 2 population pairs, and we look at, in this example, this colored surface is the likelihood density of the 2-parameter model, so they're diverging at different times. and this white line here might be a little hard to see, but that is the likelihood of the constrained model, the one-parameter model. And MS Bayes uses continuous uniform priors on the divergence times, which is represented by this hard-to-see red dotted line around the perimeter here. And so it looks like we should prefer the two-parameter model in this case, but in fact, the one-parameter model actually has a higher marginal likelihood, even though it's not explaining the data very well. And the reason for this is it has to average over a much greater parameter space that's characterized by low likelihood but relatively high prior density. And so it has a very very small marginal likelihood. And so what we're essentially doing by using these priors is saying a priori, we're telling the method, if there's any chance that a simple model can explain our data, tell me that that's the correct model. And in a lot of inference frameworks, that actually might be desirable, just not in this biogeographic context. So if we look at this from a one-dimensional perspective, one way of potentially solving this is simply to use more flexible parametric distributions on divergence times, in this case a gamma. And so it allows us to still capture our prior uncertainty about parameters, but avoid placing a lot of prior weights in regions of low likelihood. Okay. So next, why take a different approach to modeling the distribution of the divergence times? Well, MS Bayes uses a discrete uniform prior on the number of divergence events. But there's many more ways we can assign our population pairs to an intermediate number of divergence events than to either very few or very many. And so if we look at the average prior probability of any divergence model in these classes, what we actually get is the strongly U-shaped prior distribution. And we already talked about how these divergence models here with many divergence time parameters, up to 22 here, likely have very poor marginal likelihoods due to the uniform priors of the divergence times. And so if you essentially just mask those out, what we're effectively doing is putting a prior density that places almost all of the mass on very few divergence events. So given this, it's not that surprising that MS Bayes is behaving the way that it does. And so one way of potentially solving this problem is to use a flexible non-parametric prior that directly puts the prior on the sample space of the divergence models rather than on the number of events, which creates this combinatorial effect. So that's exactly what I did. I used beta and gamma distributions on the continuous parameters and a Dirichlet process prior over all possible divergence models. And I don't have time to get into the Dirichlet process, but in this context, you can just think of it as a mathematically convenient and flexible way of assigning prior probabilities directly to all possible divergence models. Okay, so to assess whether this did any good, I simulated lots of data sets, and I did this under the original model, the new model, and an intermediate model that is using the U-shaped prior on divergence models from MS Bayes, but a gamma prior on the divergence time priors like the new method. So it's intermediate between those two. And I analyzed all these data sets under all three models. Now what I'm showing here is the results of analyzing 50,000 data sets in such a way that we can compare the posterior probability of the one divergence model to the true probability of that model. And what we can see here is when all assumptions of the original model are met, it does quite well. It should fall along this identity line if it's unbiased. And then accordingly, the new method is doing quite well when all the assumptions are met. However, if assumptions are violated, we're now using the MS Bayes model to analyze data produced by the DPP model. It strongly overestimates the posterior probability of the one divergence event. Whereas the DPP method is showing much more robustness here, and even when data are simulated under a different model, it still does reasonably well. I'm going to add 4 more plots here. So the 4 over here on the left, the ones we just saw. We're looking at data generated under 2 additional models, and we see that general trend of the original method is quite good when all the assumptions are met, but if any assumptions are violated, it tends to strongly overestimate the probability of the one divergence model, whereas the new method is showing much more robustness. Okay, so next, did we improve the power by making these changes? So I simulated data sets in which 22 population pairs randomly diverge across different time scales, ranging from relatively narrow time scales to quite broad ones, 5 million generations, simulated lots of data sets, analyzed them under all 3 models. So if we look at the original method, in almost all 4,000 of those analyses, it's almost always estimating a single divergence event to explain the divergences among the population pairs, even though they diverge randomly. Similarly, the new method is also struggling when those divergences are drawn from a relatively narrow time scale, but it's doing quite a bit better as that time scale gets broader. So now let's look at the posterior probability of the one divergence model in the next plots. So not only is MS Bayes inferring the one divergence model, it's actually doing so with quite strong support across the simulations. Whereas the new method, even when it's struggling, is only estimating those with relatively low probability, which is good. And then let's slide the intermediate model in between here, and what we can see is just by putting more flexible priors on the divergence times, we improve the method quite a bit, but not as much as if we also take a non-parametric approach to the distribution of divergence times across our taxa. So I also apply the new method to a data set of 22 taxa from the Philippines to see whether fragmentation of those islands during the Pleistocene might have promoted diversification. We can see that the old model is very confident that very few divergence events explain the diversity of those population pairs, whereas the new method is showing a lot more posterior uncertainty, which is expected given the richness and the stochasticity of this model, and the fact that we're using relatively little information from the data. We expect the result to look much more like this. Okay, so in conclusion, we have a new method that is showing better estimating posterior uncertainty. It's doing a better job of, it's more accurate and more robust in terms of model choice, and it has more power to detect temporal variation in divergence times. However, I'll be the first to admit, while this method is improved, it's still not great. We're still trying to estimate a very rich and stochastic model with limited information from the data, and so I expect that we'll still be quite sensitive to prior assumptions, and we should be skeptical of strongly supported results because those are likely driven by priors on our parameters rather than by the data. And just some general recommendations. For Bayesian model choice, we have to choose our priors on parameters very carefully because those can make implicit but strong assumptions about the divergence, the models that we're comparing. And also, if you use any of these methods, you should accompany results with simulation-based power analyses and assessments of the sensitivity of the results to the priors, so analyze your data under multiple priors. And I think the next step is to take a full likelihood approach to this. The likelihood is tractable. There's really no reason why we shouldn't be using all of the information in the data. And I think a fully phylogenetic framework will help as well, because you can actually estimate the relative mutation rates of these comparisons. All of the software that went into this is up on GitHub, as well as an open science notebook that details all of everything that went into this project. Lots of fantastic people to thank. And thank you, and thanks to all the organizers of the meeting and this symposium. And I'm happy to take any questions. And if there isn't time for you to ask a question, you can always email me. Thank you.
We do have time for one or two questions.
Yeah.
So if the divergence times are random, and the space something that says there is one and it gets strong, whereas this model gets strong. What happens if they're not random and there is one? Would this model overestimate the number of divergences? Is that something you expect?
It tends to do a pretty good job, but again, we'll estimate it with lower probability. And I think that's just a reflection that this is a really stochastic parameter-rich model. We kind of expect that. So we're using an approximation that's simplifying our alignments down to relatively few summary statistics. So we really shouldn't be that confident in any particular scenario. But that's very good. Any other questions?
Okay, well, thank you.